noir plasma chrome bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.
This website makes learning so easy. I found all my resources here.
Like what you share.
The reflex arc begins. Synaptic transmission relays the signal. Neurotransmitters diffuse across the synaptic cleft. This activates the muscle contraction, crucial for diagnostic neurophysiology.
Reflexes protect us from harm. Bypassing the brain entirely, they operate through localized spinal circuits. This indicates a healthy nervous system. Everything looks completely normal.
Stimulus detection initiates the sequence. Information processing unfolds within the gray matter. Action potentials travel along the myelinated axon. The efferent signal reaches the target organ. This induces contraction in skeletal muscle.
Autonomic reflexes regulate visceral organs, from the pupillary light reflex to protective cough and sneeze mechanisms.
At the cellular level, simple monosynaptic circuits involve direct synaptic connections between IA sensory afferents and the spinal anteriorons within the spinal indivision.
Inside the spinal cord, reflex arcs rely on chemical synapses. Electrical signals become chemical messages. Vesicles release neurotransmitters. They bind to receptors, triggering a new impulse.
A synapse comprises three regions: the presynaptic axon terminal, the extracellular synaptic cleft, and the postsynaptic membrane packed with receptor proteins. An action potential arrives at the terminal. Voltage-gated calcium channels open up. Calcium ions enter and trigger snare complex assembly. Release neurotransmitters. They bind to postsynaptic receptors. Binding opens ion channels. Cation influx produces an EPSP. Anion influx produces an IPSP. Enzymes degrade transmitters, clearing the synapse.
Two distinct synapses exist. Chemical synapses have a wide gap. Electrical ones connect directly. This allows ions to flow directly between cells.
Synapses transmit signals between neurons. Chemical ones use neurotransmitters. Electrical synapses allow direct ionic flow. This makes them virtually instantaneous, while chemical paths are highly modifiable. Descending motor pathways modulate spinal reflex activity. Corticospinal tracts exert neurons innervate contractile ends of spindles. This regulates reflex responsiveness.
The examiner applies a brisk strike, creating a rapid stretch. This deforms intrafusal receptors. Be applied consistently. Voluntary tension can damp the contraction.
Tapping the chin stretches the masseter muscle. Sensory neurons carry this impulse to the pons, synapsing directly with motor neurons, causing the jaw to jerk shut.
The biceps reflex tests the C5. Tapping the tendon stimulates muscle spindles. Impulses travel via the musculocutaneous nerve. This activates motor neurons, causing a quick contraction. Testing the brachioradialis reflex taps the C5 and C6 segments. The radial nerve carries the impulse to the spinal cord. A quick contraction follows, confirming normal function.
Testing the triceps reflex assesses the C7 and C8 spinal segments. The radial nerve carries the sensory impulse to the spinal cord. A motor signal then causes the tricep muscle to contract.
The patellar reflex targets the quadriceps. Tapping the tendon sends a signal. It travels via the femoral nerve to the lumbar spinal segments L2 to L4, causing the muscle to contract and kick. Testing the Achilles reflex. This tests the tibial nerve and the S1 to S2 segments. The impulse travels to the spinal cord. This causes the calf muscles to contract, resulting in a visible plantar flexion. Superficial reflexes are polypsych responses, like the abdominal reflex moving toward the umbilicus, and the plantar reflex, tested on the lateral sole. A normal response is flexion of the toes. Stroking the inner thigh stimulates the L1 to L2 spinal segments. This causes the cremaster muscle to contract, elevating the testicle. Its absence is a key indicator of testicular torsion.
Evaluating osity. Asymmetry across joints, evidence of pathology. Documentation relies on the 9 scale. Asymmetry often provides stronger evidence of focal pathology. Just relax your leg for me. There is no response, which we grade as a 0. This indicates a complete interruption of the reflex arc. Grade 1 is a small trace reflex, present only with reinforcement. This hypoactive response suggests potential nerve damage. A grade 2 response is considered normal. A grade 3 reflex is brisk. It is in the upper half of normal. This may indicate mild disinhibition, but it can also be physiological. Notice there is no clonus.
This is a grade 4 reflex. Notice the hyperactive response. It indicates severe loss of descending upper motor neuron inhibition.
Grade 5 sustained clonus is elicited by an abrupt stretch. This indicates severe pathology. It shows definitive disruption of the corticospinal tract.
Upper motor neurons descend from the brain. They modulate the spinal anterior horn cells. Lower motor neurons extend directly to target muscle effectors.
The corticospinal tract is the primary pathway. It conducts impulses from the brain to the spinal cord. An interruption, such as a stroke, blocks these signals. This leads to a loss of voluntary motor function.
Let's compare upper and lower motor neuron lesions. The site of injury for UMN is the descending corticospinal tract. For LMN, it's the anterior horn cell. UMN lesions cause spasticity, while let's differentiate upper versus lower motor neuron lesions. UMN lesions show a positive Babinski sign and hyperreflexia. LMN lesions cause hyporeflex wasted muscles. Check the parameters.
Let's differentiate motor lesions. UMN shows spasticity. Clonus is present here. LMN has fasciculations. Atrophy is severe. Iridism prolongs reflex relaxation. This is due to delayed calcium uptake. Hypothermia causes generalized hyporeflexia. In diabetes, nerve damage causes early reflex loss.
The reflex arc is a model of neural integration. It executes rapid protective movements, bypassing thalamocortical delays.
Chemical synapses facilitate these circuits. They use voltage-gated ion channels. Vesicle exocytosis releases neurotransmitters. Differentiating UMN and LMN patterns is essential.
Like what you share.
This website makes learning so easy. I found all my resources here.
Noir Plasma Chrome Bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.